Good afternoon, everyone. Welcome. My name is Suerie. I'm a professor of international relations and co-director of the Global Health Center at the Geneva Graduate Institute. It's my great honor and pleasure to welcome you all to this session where we are going to be hearing from one of the pioneering leaders at the front lines of genomic surveillance. I'm really excited and honored to be sitting next to Dr. Christian Happi, whom I'll introduce to you in just a moment. What I wanted to do is to also let you know that you can follow the conversation, contribute to the conversation at #GESDA Summit. And welcome to those of you who might be joining us online and following us online. Please do send us your comments and questions using the hashtag. We are going to have a fireside chat for about 15-20 minutes, and then I'll open up the floor for your questions and comments for Dr. Happi before we wrap up in about 30 minutes' time. So without further ado, I'm really, really excited to be sitting next to one of Time Magazine's 100 most influential people of 2025. I would say just one of many, many honors that Professor Happi has earned over his very illustrious career. He is a professor of, I've got to make sure I get this right, molecular biology and genomics, and is also the director of the African Center of Excellence for Genomics of Infectious Diseases at Redeemer University in Nigeria. He's world-renowned for his cutting-edge work in genomics analysis and surveillance, and in particular, helping teams on the ground and countries better respond to and get outbreaks of infectious disease under control. And very excited to hear about some of the recent changes in this field, where you see it going, and some of the big political and economic questions that we are wrestling with. But let us begin first by getting to know you a little bit better. So Dr. Happi, I'd love to hear a little bit more about what drew you into this field. How did you become who you are today in terms of a pioneering scientist helping the world deal with outbreaks of infectious disease?
Yeah, thank you, Suerie. Thank you for the introduction. I think Christian Happi is my name. As you say, I'm at Redeemer University. And I think I got into this field from what I call life experience. So I was born and I grew up in Cameroon, and in the tropical forest region of Cameroon. And as a child, I had, I would say, uncountable bout of malaria. And at the age of 8, 9, I had one of the worst episodes of malaria that I can recall. I was coming from a very humble background. My mother we couldn't afford a car or cab or a bike to go to hospital. So my mother was backing me, carried me on her back and took me to the hospital. On the way back from the hospital, she would walk and she get tired. And she got tired, and then she decided to rest, and I was leaning against a mango tree. And under the mango tree, I asked her, because I couldn't stand, I asked my mom, why am I this ill? And she told me, the doctor said you had malaria. And my next question to her was, why is there no cure for this disease? And she said she doesn't know. And the last thing I told her was, when I grow up, if this disease is still on, I will find a cure for it. That, at that age, was one of the things that was driving me. And a few years later, in my biology class, I saw a cartoon of Watson and Crick. These were two biochemists. And the cartoon was saying that they discovered the DNA. And then my biology teacher told me the DNA is a molecule of life. Everything that has life, has DNA. If you take the DNA out of anything that has life, then that thing dies. I made a connection at the time. Malaria parasite and in the DNA, then I must take out the fighter from malaria parasite. That was my thinking at the time. So I graduated from high school with flying colors. And as a biology student, I was actually directed to go to medical school in Cameroon. But because of that, the power that I saw behind Watson and Crick, who were described to be biochemists at the time, I did something that was unprecedented. I didn't ask permission to my parents. I didn't ask anybody's permission. I refused to go to medical school, and I went to register in the biochemistry department because I was emulate it. I wanted to emulate what Watson and Crick did. So I graduated from the university and looked for a program that was looking at understanding the genetics of infectious disease, and specifically malaria. And I went into that program to learn about genetics and malaria. I did a bit of work, and I realized that one way to stop malaria was to use the genetics to create a vaccine. And I do remember then Professor Ian Tilley in the University of Melbourne had a very elaborate program on developing a malaria vaccine based on apical membrane antigen 1, AMA1. I wrote to him, and I showed my interest, and he gave me a full scholarship to go to Melbourne. I went, got my visa, everything, but I was wandering within my university after my master's, and then I saw an institution, say Postgraduate Institute of Medical Research and Training. I walked in there, I wanted to find out what they were doing, and I met a professor who was heading a program there, say he was working on understanding, that malaria parasites are not responding to drugs anymore, and it's killing more children. And I told him about my interest in developing a vaccine for malaria. But he gave me an advice. He told me that, well, malaria vaccine is a long shot from where we are. Maybe in our lifetime, we may not be able to see a malaria vaccine. But he also told me that I see in you someone that is in a hurry, someone that has fire in him, and someone that wants to solve problems. Maybe you should rather think about understanding mechanisms of resistance and maybe developing countermeasures for malaria that kill children, that is a faster way of addressing the problem that you want to address. And I made another crazy decision where I sat down and I said, oh, this guy is right. And after 2 days, I changed my mind again. I wrote to the professor in Australia, and I told my colleagues, well, I don't think I'm going to Australia anymore. I think this problem is more urgent, and then we can solve it this way. Of course, I started our program, eventually designed something that was kind of crazy, and sent it for a conference, and then I met my mentor at Harvard. That's why she took me over to Harvard, and then I went there, ran this program, made quite a number of discoveries in the field, understood that mechanism, described it very well, and also start thinking about how we could reverse it. So this is how I got into the field. But then you move forward, and while in the US, I grew up, you rise a rank, and then every time I'm in my lab, I will just realize you open the news, and then you realize one disease is killing people in Africa, and that disease was Lassa fever. And I was like, how come? I read about it. That was 2007. And I'm like, a disease that was described in 1969 for the first time in Nigeria. There's no diagnostics. Anytime, there's an outbreak, 90% of the patient die, simply because there's no diagnostic, there's no lab. And also because when the sample gets sent to Germany, I think it was a Bernard Notch Institute or something like that, by the time they get the result, probably a few weeks or months later, 90% of people are dead. I was like, it's not possible that this should be happening at this point. So I quickly went on and then read a bit about it, developed the first molecular diagnostic for it done in country, went from the US, went back to train people, and then I could see over time, in little run of time, the case fatality rate by just introducing that diagnostics, we cut down the case fatality significantly. You talk about 90%, we came down to about 26.7%. At that, I started seeing that, making those kind of interventions could be very useful and could be very helpful. But clearly, my background in genomics and genetics gave me also an opportunity here. This was a disease that was a neglected tropical disease. And I realized that we could accelerate processes by actually understanding the genetic makeup of the virus, because that was a major limiting factor. If you don't understand the genetic makeup, how do you quickly accelerate vaccine development? So I went on actually to create to do the first genome sequence of that virus. And by doing that, we found a lot of things, not only understanding, the ancient nature of the virus, first described in 1969, but our data actually showed that the virus was much more older, 1,181 years, was circulating within Nigeria for 600 years before it moved out. But such information accelerated vaccine development. So within 5 years, we went from having no diagnostics, but to move to have a vaccine or vaccine candidate that actually are undergoing clinical trial now. So just give you a kind of background there.
So thank you. I would really love to have you kind of walk us through some of the really dramatic changes in genomic sequencing activity and capacity in Africa over the last 10, 15 years. But I want to draw, to me, a few interesting takeaways from your story, how you got into all this. I think first, the incredible dedication of mothers. I think that's clearly something that if she hadn't carried you to the hospital, you may not be with us today. Second, the incredible importance of high school biology textbooks, which inspired this scientific spark of curiosity. But last but not least, you are clearly a man in a hurry. And you really, speed is of the essence. You want things to move faster. And I think this is a nice way to segue indeed into, would you describe for us the landscape? What was the infrastructure and the capacity to do genomic sequencing in 2010, 2011, when you finished up your training at Harvard? You went back to West Africa. What did it look like? How has it changed since then? And how has it affected responses to crises like Ebola, Marburg, Lassa, COVID? I mean, so many different crises that you have played a really key role in helping to address.
Yeah, thank you. I think in 2011, when I made that decision to actually go from Harvard back to Nigeria, I think I went and I was telling my colleagues I was going with a mindset to set up a genomic facility in Nigeria. And everybody at Harvard told me that, look, Africa wasn't ready for that technology. Imagine 2011, genomic was still pretty much in its infancy. That Africa wasn't ready for that technology. You don't have everything that is needed. You don't have power. You don't have water. You don't have anything. So maintenance is a problem. But I went with that mindset, and I was able to convince an entity like Illumina. And I told them that, look, get me a sequencer, and we're going to change the world. And fortunately, I think the CEO at the time heeded to it. And then we got the very first MySeq sequencer on the continent. So there was nothing. There was no infrastructure. There was no human resources. Then we really started from the ground.
That was just 15 years ago, 14 years ago.
Yes, there was none. That was the very first sequencer on the continent.
And today?
But then barely, 6 months after I was given that sequencer, I was really thinking about continuing to do some of this work. We had the Ebola outbreak in West Africa. And of course, over the years, I've realized that the way we respond to outbreak on the continent, on the African continent, was absolutely inadequate. Every time there was an outbreak on the continent, experts will come, in quote. They will try to work with the countries. Samples will fly back to some lab in Europe or somewhere in the US. 2 years after, they will tell you, what caused that outbreak that killed almost everybody was this particular virus. And I found that wasn't really the way to do things. I found that, I was telling myself that if we can do this in real time, characterize in real time, the doctors in Africa do have enough experience to deal with these things. All they need is to get information. And when Ebola started, I had a series of ideas that I thought if we deploy, that could help change the way we respond to epidemics. And I deployed that. The first thing we did was to do real-time genomic sequencing. And we did that on the ground. And also the second thing we did was to do open data sharing, open data access. We sequenced the first 99 Ebola genome at a time and made it openly available to the world. So pioneering open data sharing, open data access. The third thing we did is looking at the landscape. Diagnosis was a big challenge. And it became very challenging for people to, test and respond. People that had malaria will go to Ebola triage center, get infected there, and then take the virus back home. But we realized if you speed up diagnostics, if you get a rapid diagnostic test, you could cut down the diagnostic time, you could reduce, you could make testing in your real time. And that was the third thing we did. We went from genome sequences and created the very first rapid diagnostic test that was approved by WHO and then the US FDA under emergency use authorization within a very short period. That really became very catalytic. So because people could see the whole concept of translational genomics from genome sequences to product. And the introduction of that test flattened the curve, right? Then we also observed one thing in the process. One particular variant, even though introduced later than other variant that was circulating, takes over and masks every other variant. So the question was, what was making this variant, seen more than others? That was when, again, through genome sequencing data and then, functional analysis, we realized that this particular variant had mutations, about a large number of mutations, 327, but you could see 21, very conserved. And then we could actually relate those mutations to infectivity. So we established that this particular variant, SL3, actually was, the mutation in that particular variant were associated with increased transmissibility of the virus. That's when we discovered the concept of variants of concern during an outbreak.
Which unfortunately we all became very familiar with during the COVID crisis.
Exactly. So you could see that all of the pillars that we established in 2014 became what the world used in 2020 to respond to COVID. But then to demonstrate the power of this approach, to epidemic response, we tested this and fully deployed that in Nigeria. Nigeria had its first coast case of Ebola in June 20th. Unlike other African countries that were sending samples out or taking time to do, within hours, we knew and confirmed that we had. And by the third day, we had a full genome sequence. We knew exactly where people were. We established transmission chain. We deployed this whole chain that I'm talking about, all these instruments. And we demonstrated that it works, and it works fantastically well. Nigeria, a country of 230 million people, with a disease like Ebola emerging in a city like Lagos, 25 million people. The country contained this in 92 days with only 20 cases and a death.
This is an incredible success story, and I know you have many, many really fascinating success stories, but I want to ask one more question because I see, maybe I have time for two more questions. I think one of the issues that came out of COVID and also the Ebola crisis was what happens when scientists such as yourself share data openly, very valuable data for public health purposes, very valuable data sometimes for economic and business purposes. And we saw, of course, during the COVID crisis that many countries, African countries, were sharing genomic sequencing data quite early, and yet we're very much back of the line for vaccines and drugs and diagnostics. And today in Geneva, over the last few months and over the next year, you have diplomatic negotiators who are working very hard, trying to come up with better rules so that this does not happen again. So I guess my question for you is, what would you say to those negotiators in terms of, what do you think is fair for researchers like yourself to receive in terms of benefits in exchange for making that data available? What is it that researchers should receive? What is it that countries should receive? How do we try to inject more equity and fairness into the current genomic sequencing ecosystem, if you will?
Yeah, I do think that when in 2014 we actually pioneered open data sharing, open data access was actually for global health equity. It was actually to help accelerate vaccines, drugs, and diagnostic development, but with the mindset that when some of those things are developed, they should think about the benefit of the people that actually contributed in providing those information. So, and I believe that, it's only normal and it's only fair to work together and then look at the world as a place where there should be some level of equity and fairness. I think it is important for people that are accessing the data to use the data knowing fully well that people contributed the data. Not only the data, but they add metadata. Without the metadata, you can do much with them. So, and giving back actually brings some level of fairness and encourage people to actually put more. But a situation whereby the data are taken, diagnoses are created, vaccines are created, even the countries that we have that contributed significantly with those data, they can have access to even IPs or TRIPS, at least even to use it to improve themselves. That becomes very selfish. And that's the reason why during COVID, for instance, some countries, deliberately started refusing to share data because there was no benefit. African countries, even though having discovered the Beta variant and Omicron variant, who helped the world to refine vaccines, were deliberately refused, to buy vaccine even when they had the money. So, and I think we should rather look at the world as a place where we can actually live together, where we could protect one another. Otherwise, as I said yesterday, we as human are endangered species. If people keep holding back on the data, then it means that, of course, people will not know what is circulating. The second part of it is not even just about sharing the data. Stigmatization is a big problem. You find something that is very important for public health, for public health. You share the information, but then the next thing is that you stigmatize against. It doesn't just make sense. We should rather think about doing things that are a bit fair and equitable. And one thing, for instance, that we use our platform to bring about fairness and equity was the whole story of mpox. Mm-hmm. After COVID, the world had a huge spread of mpox, and then it spread from Europe to the rest of the world. There was this natural narrative to say that, oh, it came from Africa, it emerged from Africa, but we use the data in our lab, showed clearly that it emerged somewhere in Europe. I don't want to mention the name. But then also the narrative where they say, oh, there were different lineages or clades. People were very brave to call some clades Central African clades. They were very brave to call some other one West African clade. But we have clear demonstration with data and evidence that this clade emerged from Europe and spread. So my question to WHO and then the international community of taxonomy of viruses that is we're going by geographical taxonomy. What do we call this particular clade? Of course, it makes some people uncomfortable. We don't want to call it a European clade. We should call it European clade because it's emerged from here. Of course, we eventually reached a common ground, which was have a taxonomy that is more neutral and less stigmatizing. That's where you saw the origin of talking about clades, 1, clade 2, and then clade 3. That's basically, so if we could work in a world where there is, fairness, where there is some level of equity in the way we treat people, information, data, I think we will all make progress, and then the world will be a better place to be.
Thank you. I'm sure many negotiators will be very interested in hearing your message. This is really the last piece of the puzzle that needs to be put in place before the pandemic treaty can be open for signature and come into force, so that hopefully we can indeed have more effective and equitable responses to future crises. And I think just hearing about your trajectory, we are reminded of these sort of non-stop march of pathogens that we are constantly wrestling with. Very, very different pathogens, all creating very, very different kinds of crises. What I want to do is ask you about 50 more questions, but I'm not going to do that. I'm going to open up to the floor because I see the clock is already ticking for questions or comments from our audience today. And please identify yourself briefly, and then we'll have you pose your questions. Yes, let's start with the young lady here, and then the gentleman on this side.
Hi, thank you so much for the story you told us. So my name is Daniela Doda, and I work for the Lichtsteiner Foundation here in Switzerland. So my question was, in the journey when you came back from Harvard, and you wanted to bring genomics to Nigeria, to Africa, what was the cultural acceptance there? Like, did you find it welcoming, or what was your experience when you talked to the common people, and not the scientists and the policymakers?
Yeah, thank you. I think at the beginning, people don't really understand what you're saying. Genomics is a new vocabulary. They don't see the relevance. It's another researcher doing this research. But then I think what is important for policy makers, and I've seen the case in Nigeria, is actually evidence, the data, the fact. So in the case of Nigeria, at least they were able to see this in action. They could see the value, the power of genomics. And then they could see, while other countries were losing 11,000 people, they ended up with only 8. And that they could see because the data given to them in real time, you could identify transmission chain, you could identify hotspot, you could contain it. So you actualize these things. And I can tell you what, I work in a private university. And in Nigeria, the government did something that they often don't do in Africa. Go to the World Bank, get a loan, and these are loans in tens of millions of dollars, and give me all the money. And I said, look, just do the work. So I think evidence is what matters. And with that, people get to, they accept those things, they become used to it, and we've actually used that as a way to propagate this approach across Africa. Right? So you saw the way Africans responded to COVID, significantly. It's simply because we were able to actually sequence COVID in 48 hours and also openly share our protocols to all African countries and also build a capacity within. So that really actually helped, in great ways, to show that it works. all those variants that you see discovered is actually based on modification of the protocol that we share. So I think it shows the power behind it and then the evidence that it works.
Thank you so much. We have, yes, please, let's take maybe 2 or 3 comments, and then I'll turn back to you.
My name is Maxim Baines. I'm a high school student here on behalf of the Villars Institute. And you mentioned earlier the need to make data more open source. So my question is, what kind of global governance framework do you think should be established, if you think one needs to be established, to have transparency and data sharing and genomic surveillance in both the public and private sectors?
Yeah, thank you. Wow, what a bright question.
A few,
okay,
so that we get a few more inputs. We have a gentleman over here. Thank you.
Thank you, Professor Happi. That was fascinating. I'm keen from the Wellcome Trust perspective to understand what we should be doing as a health research funder to address the ability of researchers across Africa to have access to the machines and the supplies that they need to do their work. In your view, should Africa be developing a rival machine to Illumina, or should the emphasis be on ensuring that Illumina is supplying what you need?
Okay. Back to you.
Right, I can start with that question. So I don't think that we need to, you don't want to reinvent the wheel, right? You don't need to say you're developing a rival machine. But we can work with Illumina, for instance, to make their technology better. And that's what we've done. So we have an agreement with Illumina. We have early access to technologies. And I can give you some instances where Illumina developed a technology and then we demonstrated to them that was completely flawed. We agreed and we developed the technology, and the technology co-created, co-developed it, and it works better. But now, in order for Africa then to show that the talents are there, things are there, and by the way, actually the genomic sequence data that we're generating is not just for epidemiology purpose. I mean, if you check the literature, we created the first CRISPR-based, used gene editing technology diagnostics for an infectious disease was Lassa fever. We've done that for many other diseases. But the point here is for Africa to actually just help not only itself, but also the world, is agencies like the Wellcome Trust to do, to empower the African countries and the African institutions to do it. Give the resources to those countries. They are accountable, they can do work, but a situation whereby I do understand that, they have to be dependent. I'll be an appendage to an investigator in the UK, and then 70% of the resources remain in the UK. I don't think that's correct. I don't think we're doing a good thing. Let's give them the resources, and then you will see how they will transform the world. That's what I would say, with regard to that. Then the question I think I want to say very vous êtes très brave. It's very smart to see a high school student asking that kind of brave question. And I want to say that, your question is very timely. Transparency is very important. I think one of the major challenges that we're facing now and maybe going forward is the fact that there's no transparency in the way the data are accessed and in the way the data are used. And I think one framework that could be required is actually a situation whereby the data owners, remain the owners of their data, and then those that want to access the data can work with the data owners to access the data. That way you create collaboration. That way you create, you can co-create in terms of addressing the research questions that somebody that wants to address the data wants to do. And you can also create what we call TREs, which is trusted research environment, where people can all come together, and collaborate on work and collaborate on things. That will bring some level of equity. IPs are co-shared. People will be more than willing to share and collaborate. I think if you do that, system is good. But a situation whereby you are depositing the data and then somebody just come and grab the data and goes with it, I think does not promote that kind of collaboration and then that kind of equity, and therefore people are reluctant, to put in the data. I think that's the kind of framework that I suggest we do, and then that will be very helpful to everybody and create a win-win for all.
Thank you so much. And this is probably a fantastic note on which to wrap up our conversation. For me, there's a few key themes that really jumped out from the conversation. One is the incredible speed and the extent to which this increase in genomic sequencing capacity and analytical capacity has been a game changer in the way countries have been able to respond to outbreaks. Very, very impressive and exciting. The second, I would say, is the importance of indeed continuing to invest in building up and further developing that capacity. And I know this is a big passion of yours, and I didn't manage to squeeze in a question earlier, but very impressive that you have now trained scientific researchers from 53 out of 54 countries on the continent, and I'm sure that effort will continue. And I think the third is the importance of these global rules, right? If we want to indeed build more equitable systems for the sharing of scientific credit, for sharing in the fruits of scientific knowledge, this is where diplomacy, including processes, of course, happening here in Geneva, really have to meet the moment and play their role. So with that, I knew our time would fly by. I really want to thank all of you for coming and joining us today and for the excellent questions that you've put on the table. I want to thank you, Dr. Happi, for all your work and for joining us for this fireside chat. I've been instructed to also remind you, you can continue the conversation at hashtag GESDA Summit. There will be a survey for this session with a, oh yes, QR code. Please fill that out and provide us your feedback. You have the full program for the rest of the summit available online, and there will be another session on pandemic preparedness tomorrow morning at 9:15 in Auditorium C. So with that, please join me in thanking Dr. Happi, and I wish you a good rest of summit.